You everyone for coming to this talk, and thank you to the UCL team for inviting me to give this talk. I will be continuing describing the Neuropixels Opto probe as Carolina started, but this time in terms of a different application for the probe, which would be for optotagging different cell populations during electrophysiology. So, as Carolina described, an important thing that is useful to know during an electrophysiological experiment is the cell types of the neurons that you are recording, because different cell types are known to have very different effects on behavior, play different roles in circuits. But currently, the best known way to identify cell type populations during an electrophysiological recording is this technique that I'm sure many of you are familiar with, called optotagging, where you induce expression of a light-sensitive opsin in a cell type of interest, and then later identify that cell type during an electrophysiological recording by their low latency responses to laser pulses. So in this example experiment, the authors of this paper induced expression of channelrhodopsin, which is a blue-sensitive opsin in midbrain dopaminergic neurons, and then identified them during the electrophysiological recording by their responses to these blue pulses of light. However, despite optotagging being, the sort of the current gold standard for identifying cell types during electrophysiological recordings, it's not an experiment that's very commonly done. Simply because it can be kind of a challenging experiment to pull off. So in addition to the fact that you need transgenic animals, light delivery can be an issue for many of these experiments. So for instance, the light delivery needs to be in the same location as the recording. So if you're using either an external surface stimulation or fiber implant, if you simply are not directing the light at the same location that you're recording from, you're not going to see responses. If you are especially if you're recording from deep subcortical regions, if you're using a fiber to deliver the light, you can cause a lot of tissue damage to surrounding brain areas. And in addition, this technique often creates very large photoelectric artifacts, which, especially if they saturate the physiology, can greatly decrease the quality of your data. And so we have created this Neuropixels Opto probe that should be able to address all of these issues. The light emission sites are on the shank, so you do not need to worry about the intersection between recording sites and light emission sites. And because of the profile of the probe is roughly the same as the Neuropixels 1.0 probe, so there's no additional tissue damage that isn't already caused by just the insertion of the probe for recording. And because of the relative low power of these emission sites and the fact, as Carolina showed, that the light is directed away from the recording sites, the photoelectric artifacts that we see from these probes are pretty minimal, especially compared to surface stimulation that would be needed to drive the same sorts of responses. So at relatively higher stimulation from the opto probe, you do still see some photoelectric artifacts, but they are they are not saturating, and they are basically entirely removed by normal pre-processing steps. So the quality of the data is greatly improved with these Neuropixels probes. So in our case, we are using these probes to optotag different cell populations in the brain. So in this example experiment, we used a genetic strategy while recording from dorsal striatum. We used a genetic strategy to induce the expression of cochir, which is a blue shifted opsin, in striatal D1 neurons, and carmine, which is a red shifted opsin in striatal D2 neurons. So in this case, the D1 and D2 neurons are a largely non-overlapping populations of neurons within dorsal striatum. So we are tagging two relatively unique cell type populations simultaneously using the Neuropixels Opto's the fact that it has these two different color light emission sites. And here you can see a couple of units that we managed to pull from this experiment. So we have these first two units, which have strong low latency responses to light from emitter number 3. So both of these would be two different D2 neurons, and then units 3, 4, and 5 have these low latency responses to the pulses of blue light, indicating that these are 3 different D1 neurons that we recorded in this experiment. And in fact, in this particular experiment, we found that of the portion of the probe that is in striatum, we were able to assign a cell type to the majority of the units that we recorded. So here I'm plotting by depth along the probe, I'm plotting every unit passing quality control that we recorded in the session. And of the portion of the probe that was in dorsal striatum, you can see the majority of these have a cell type label assigned to them based on their responses to the laser. So obviously we were able to do this with the D1 versus D2 neurons because together they make up about 95% of units in dorsal striatum. But this is still like pretty amazing yield compared to other optotagging experiments. When we look at the spatial distribution of tagged units, when we align them to the emitter driving laser responses in these units, we find that they are slightly, or I guess not really slightly, but they are biased to be below the emitter that is driving the responses, which if you recall from the plot Carolina showed of the light profile from the light emitters makes sense because they are the light is sort of directed downwards from the emitters, so it makes sense that the cells being driven by the emitters tend to be below the emitter. However, if we instead align the units to just the nearest emitter, we find that the distribution of tagged cells is pretty close to the distribution of untagged cells, especially in the Y direction. The distributions are not significantly different from each other. In terms of the X direction, we find that the units that are tagged do tend to be clustered a little bit more towards the center of the shank compared to the untagged neurons, which have this a bit more of a tail around the edges, which does sort of indicate that there is some subset of units that are like right on the boundary of where they can be of where the data can be collected. So overall, the majority of units that are in range to be picked up by the electrical sites of the Neuropixels Opto probe are also in range to be stimulated by the light emission sites, although you might miss some units that are like very, very far away from the probe and like are just barely being collected. There are a couple of limitations with the current design of the Neuropixels Opto probe. So for instance, you might have noticed that especially for the units that are responding to the blue light, they were being driven by more than one emitter, and that we believe is because of some light leakage problems with the current Neuropixels Opto design, but that should be fixed in the next iteration. There's emission sites there's only 14 emission sites in each color, covering the bottom 1.4 millimeters of the shank. But later iterations of the probe should have, I believe, 48 of each color, covering a much larger area of the probe. Another kind of funny problem with the current probe is that it's very, very large compared to other Neuropixels probes. So for the experiments that I showed, we were basically completely limited to only recording from 2 probes at a time because it's just not feasible to insert more of these than that into a mouse brain at the same time. But future Neuropixels Opto probes will have reduce the footprint to be about the same size as the current 1.0 probe. So these probes, it would be feasible to use these probes in higher numbers, higher densities to both optotag and do optogenetics experiments across a broader variety of brain regions. Sorry, it was 44 sites per color. So the current timeline would be that next year we should have some of these alpha probes, which have the smaller form factor and larger number of emission sites. And then perhaps by 2028, these opto probes will be commercially available for purchase from IMEC. So I want to thank you to everyone at the Allen Institute for Neural Dynamics that helped support these experiments. And then obviously everyone in IMEC, Genelia, UW, UCL who are part of the Neuropixels Opto consortium and aided with the development of these probes.